Hi, I'm Jamie O'Reilly, and I'm going to summarize the main points from this paper about mismatch responses in mice, published in the European Journal of Neuroscience. Mismatch negativity is defined as a change detection response. It's observed from human EEG during an oddball paradigm. The interesting thing about MMN is that it seems to be different in patients with schizophrenia and other mental illnesses. So the purpose of this study was to characterize the mismatch response in mice, in the hope that this can be used to help understand what causes it to go wrong in different diseases. Mismatch negativity in humans is observed when they're anaesthetized or conscious. So in this study, two separate groups of mice were used, an anaesthetized group and a conscious group. The oddball paradigm included increasing and decreasing oddballs, shown in red and blue triangles. Three separate versions were played with duration, frequency, or intensity deviant stimuli. There were 800 standards, shown here with white circles, and 100 of each deviant, so in total there were 1,000 stimuli. Three versions of the many standards control paradigm were used, with 10 different duration, frequency, or intensity stimuli, each played 100 times. This serves two functions. It can be used to study the auditory response to different physical properties, and also to view the response to stimuli that are physically identical to the deviant, but in a different context, without being preceded by repetitions of the same standard stimulus. Here A shows that the experiment was conducted in blocks, with the oddball paradigm first, then the many standards paradigm in the second block. B illustrates the main observations. Essentially, standard and deviant stimuli in the oddball paradigm produce different auditory responses due to their physical properties, and differences between oddball and many standards paradigms were due to a systematic reduction in response amplitudes, presumably caused by repeated exposure to sounds, and we can call this auditory habituation or adaptation. C shows different waveform comparisons that were performed and how these were interpreted. Classic mismatch responses were computed by subtracting the standard from the deviant in the oddball paradigm, while controlled mismatch responses were computed by subtracting the control deviant from the oddball deviant. There wasn't any notable difference between the controlled mismatch response and the controlled standard difference waveforms. Both appeared to reflect auditory habituation, so for the rest of these slides, I'm just going to focus on classic mismatch responses in mice. Here are the duration many standards responses from anaesthetized and conscious mice. We can see that the waveforms from the two groups are quite different, but there are some similarities. The first feature in both is a negative amplitude peak at approximately 20 milliseconds. This basically has identical amplitude for all of the stimuli, which had the same frequency, intensity, and interstimulus interval. After the conscious group shows a more dynamic alternating response with several positive and negative deflections. It's also clear that both groups exhibit offset responses that are directly proportional to stimulus duration. Here are the auditory evoked potentials on the top and classic mismatch responses on the bottom. We can see that offset responses fundamentally shape the difference waveforms, with the standard stimulus offset response causing a negative peak at approximately 125 milliseconds, and deviant offset responses causing positive peaks at roughly 75 and 175 milliseconds. Now we have responses to different frequency stimuli plotted up to 200 milliseconds. Higher frequencies are shown in red and lower frequencies are shown in blue. There is clearly some modulatory influence of tone frequency on the auditory response, with frequencies towards the higher end of the scale evoking larger amplitude responses. This is reflected in the frequency mismatch responses. Increasing and decreasing frequency deviance produce opposite polarity features in the mismatch response. It's a very similar picture for sound intensity. The loudness of a sound is directly proportional to the magnitude of the auditory response. Responses to quieter sounds are shown here in blue, and to louder sounds are in red. This is also reflected in the mismatch response waveforms. Louder and quieter deviant stimuli produce opposite polarity features when the standard response is subtracted from each. To summarize, mismatch responses were fundamentally caused by the inherent sensory response to different physical properties, whether this is offset response time changes with stimulus duration, or overall amplitude modulation by frequency or loudness. No evidence was found for a general change detection mechanism, and this does not agree with theories of MMN in humans, although it is the case that mismatch negativity in humans is not fully understood at this point. If you're interested in learning more, there is much more information in the article, and you're more than welcome to read it.